Welcome to the latest Novogene webinar, A Beginner's Guide to Next Generation Sequencing, Principles and Recent Developments. Today's webinar discusses the history and principles of NGS, current applications of DNA and RNA sequencing, recent developments and the future of NGS, particularly multiomics. All our previous webinars are available to view on demand on our website. A bit of housekeeping before we start. The presentation will run for around 45 minutes with 15 minutes for question and answers at the end. Please submit your questions via the Q&A button at the bottom of your screen. You can upvote a question by clicking the thumbs up symbol next to it. We'll try and prioritize the most popular questions. If we don't have time to answer your question, we'll come back to you via email after webinar. Please don't use the raise hand functionality. Just send any questions via the Q&A or message us directly if you have a technical issue. We will share the slide deck and the recording of the webinar via email. You can expect this on Friday. If you are studying and need a certificate of attendance, there will be a link in the follow-up email that will display the certificate. You can print from your browser. Finally, there will be a short survey that pops up when the webinar finishes. Please take a couple of minutes to complete this to help us to plan future webinar topics. I would now like to introduce your host for the webinar, our account manager for Italy region, Luca. Luca, over to you.
Thank you so much, Miranda. Hello, everybody, and thank you for joining our webinar entitled A Beginner's Guide to Next Generation Sequencing, Principle and Recent Developments. My name is Luca, and I'm Novogene's sales representative for the Italian market and the host for today's webinar. Shortly, I will pass over to Dr. Amadi, who will take you through the principle and recent development in NGS. But before I do that, I'm going to speak a little bit about Novogene, especially for anyone who's joining us for the first time today, and about what we can offer you to assist in the next generation sequencing journey. So Novogene was founded in 2011, and it has been growing rapidly since then. Our company mission is to advance genomics and improving life. And as you can see here, our services cover a huge range of application with real-life application from complex disease research and oncology to food and water safety. Our European Sequencing Center is based in Cambridge Science Park in the UK. Here we have our state-of-the-art lab equipped with the latest Illumina NovoSeq 6000 sequencing machines. We have always provided fast turnaround times and high-quality data, but that is set to improve even further with the installation of Falcon 2, our new intelligent sequencing platform, which I will touch on further shortly. We also have our supercomputing cluster, so we can offer bioinformatics services and store your data in the UK. However, at heart, we are a global company. We can offer services across Asia, in the US, and of course, Europe. With almost 2,000 employees and over 5,600 customers in world-class university, biotech companies, and hospitals, we can provide sequencing for the smallest or the largest projects. We provide full range of sequencing services, such as whole genome sequencing, whole exome sequencing, various types of RNA sequencing, metagenomic services, and epigenomic services. These can be applied to a wide range of research topics, from human disease, microbial cell factories, gut health, to agriculture and animal health. So the NGS market is crowded. There are a lot of good reasons why you should trust Novogene with your project. We have a really exciting development recently in the installation of the Novogene Falcon 2, our new intelligent NGS delivery platform. This new technology was developed in-house by Novogene and will massively increase the capacity of our Cambridge Sequencing Center. It will also increase the quality of the data produced as it's entirely automated and will remove any possibility of human error. With this new addition, we are now able to offer our fastest ever turnaround time for RNA sequencing, just 10 working days from sample arrival to data release. The Falcon 2 really highlights the innovation we strive for here at Novogene in an effort to keep pushing the frontier of genome research. The benefits of our in-house innovation are clear in our record in the scientific community, which we were very proud. We have been cited in over 719 research papers with a total impact factor of more than 6,120 points. Also driving the NGS industry forward with over 37 NGS-related patents. Our technologies is helping to make the NGS process more efficient and effective, all the while improving the quality of the data we deliver. The Novogene community keeps on growing. Our expert sales team speaks over 10 different languages and are located across all Europe, ready to assist in your next project. If you're not ready to send a sample yet, so you can still benefit from our educational outreach to learn how NGS can work for your project as today. We recently wrapped up the first installment of our European roadshow, where members of our sales and product teams deliver seminars about the application of NGS in DNA and RNA research. Keep an eye out in our social media for future seminars, announcements. Hopefully, we will be visiting somewhere near you soon. I would like to give you an introduction to our lovely sales team that are across all of Europe. We have the UK and Ireland team on top, our DACH region team, and our Benelux region team here. Our team has grown so much that it takes 2 slides to show them all. Here we have our Southern Europe, Nordic and Eastern European teams. If you would like to speak at your local sales representative about an incoming NGS project, please contact us via our website and you will hear back from your local sales representative soon. With that, I'd like to thank you for your attention and I will hand over to Dr. Mahdi. He holds a PhD in epigenetics and he's one of our product marketing manager here at Novogene. With that theme, he's in charge of DNA and RNA sequencing products, as well as epigenetic ones. With that expertise, he will guide you through recent developments of next generation sequencing today. Over to you, Reds.
Thank you, Luca, for the kind introduction, and welcome to everyone again. And thank you for joining today's webinar, where I'm going to talk about the principles and the recent developments in next generation sequencing. Before I start with my presentation, I would like to give you an overview of today's agenda. In the first part, I'm going to highlight the history and the current sequencing tools that are available on the market. After that, I'm going to showcase some commonly used applications for DNA and RNA sequencing. And finally, towards the end of the webinar, I'm going to give you an overview of the next generation sequencing workflow and a few ideas and few comments on the future of next generation sequencing. So to start with, next generation sequencing by definition is the process of determining the nucleic acid sequence. In other words, we are interested in determining the order of nucleotides within the DNA molecule. In fact, the sequence of the nucleotides that we commonly know as A, T, C, and G encodes the biological information that cells usually use in order to develop and to perform their function. In fact, unraveling the DNA sequence is very important and fundamental in order to understand the function of genes and the genome overall. Currently, there are different methods available for DNA sequencing, and each has its own characteristics in terms of advantages and disadvantages. If we take a look at the history of next-generation sequencing, we can see that within 20 years after the discovery of the genetic material and the DNA double helix, the first sequencing tool arrived, which is mainly Sanger sequencing. Sanger sequencing is considered to be the first generation of sequencing tools. And it remained one of the most commonly used sequencing technologies for a long time until the early 2000s, when the second generation sequencing tools were developed and commercialized. And this is mainly represented by Illumina sequencing. And soon after that came the third generation sequencing tools, such as nanopore and the SMRT technologies from Pacific Biosciences. So to recap again, the first generation of sequencing tools is mainly represented by Sanger sequencing. And if you have ever used this for your research projects, you know that you can sequence DNA fragments ranging from 500 up to 1,000 base pairs. For the second generation sequencing tools, mainly represented by Illumina, we are able to sequence fragments ranging in size from 50 up to 500 base pairs. And finally, for the third generation sequencing tools, mainly Oxford Nanopore and Pacific Biosciences, we are now able for the very first time to sequence fragments that are tens of kbs in length on average. In the next couple of slides, I'm going to highlight the different sequencing principles of the different sequencing platforms that are currently available on them. So starting with Sanger sequencing, the first step requires primer annealing and chain extension using fluorescently labeled nucleotides. The product of the step will be fluorescently labeled DNA samples that will be loaded into the instrument to undergo capillary gel electrophoresis and fluorescence detection. So what will happen at this stage is that the instrument will measure the fluorescence and identify the nucleotides that are present within the DNA sample. It is noteworthy to mention here that the very first draft of the human genome was actually developed using Sanger sequencing. Now for Illumina sequencing, the first step is library preparation, where the DNA is fragmented into small pieces to make it compatible with the platform. Soon after that, the DNA fragments are ligated to adapters to generate a DNA library. The DNA library itself is loaded into the Illumina flow cell to undergo library hybridization. Following that, multiple bridge amplification cycles will be performed to generate amplified clusters. During sequencing, several fluorescently labeled nucleotides will be incorporated into the DNA through multiple sequencing cycles. Meanwhile, the instrument is measuring the fluorescence and collecting the data. And this is how we are actually able to decode the identity of the nucleotides within the DNA molecules. For Pacific Biosciences, a similar principle can be used. The first step of library preparation includes the ligation of specific type of adapters called smart bell adapters to the DNA molecule. During sequencing, the DNA library is added into the instrument, where the DNA fragments will be sequenced using a DNA polymerase that is fixed at the bottom of the well, along with fluorescently labeled DNTPs. During the sequencing, the instrument is going to detect and measure the fluorescence signal during the sequencing reaction by the DNA polymerase, and thus is able to identify the identity and the nature of the nucleotides within the DNA molecule. One of the main advantages of sequencing using the Pacific Biosciences platform is the possibility to choose between two different sequencing modes. The first one is called continuous long reads, or CLR, that is recommended for large insert sizes. Using the CLR sequencing mode, what will happen is that the DNA polymerase will sequence the DNA fragment only once. As you can expect, since the DNA fragment is only sequenced once, any base error that might have happened during this single pass will not be corrected. On the other hand, the circular consensus sequencing, or the CCS mode, is highly recommended for small insert sizes. And what will happen in this case is that the DNA polymerase is going to sequence the same DNA fragment multiple times. As you can imagine here, any base error that might have happened during the first or the second pass of the DNA polymerase will eventually be corrected, and the product here will be highly accurate sequencing reads that we commonly refer to as high fidelity or hi-fi reads. And finally, for the Oxford Nanopore platform, the sequencing strategy is a little bit different. The technology itself relies on a nanopore protein that is fixed on a membrane. During sequencing, a single strand of the DNA molecule will pass through the nanopore from one side of the membrane into the other. While this strand of DNA is passing through the membrane, there will be a change in the ionic current. The instrument itself will measure the reduction in the ionic current and assign a nucleotide identity to it. And this is how we are actually able to determine the sequence of the nucleotides within the DNA molecule. Now, as I mentioned earlier, all of these different platforms have their characteristics in terms of advantages and disadvantages. In this table here, you can see a summary comparing advantages and disadvantages of all the first, the second, and the third generation sequencing. So starting with Sanger sequencing, we know that the advantages here include cost. Sanger sequencing is considered to be very cost-effective for a low target number of genes or genomic regions that we are interested in sequencing. In addition to that, it has very well-established workflows and very simple data analysis pipelines. However, sequencing with Sanger can be considered can provide us with low sensitivity and scalability, and more importantly, very limited sample input requirements. On the other hand, sequencing with Illumina is considered to be better than Sanger because it provides us with increased sensitivity and a higher throughput. In other words, we can generate way more data with the same amount of DNA that we use in the beginning. However, sequencing with Illumina is considered to be expensive for a low target number of genes or genomic regions that we are interested in, can be time-consuming, and more importantly, is limited due to the short reads. We can sequence only up to 500 base pairs and not beyond that. Now, for the third generation sequencing tools, and starting with Pacific Biosciences or PacBio, the main advantages here include the ability to sequence long reads, high accurate reads with the CCS mode, and for the very first time, direct detection of epigenetic modifications. However, sequencing with PacBio is considered to be expensive, requires large amounts of starting material, and more importantly, there is a high error rate with the CLR. Finally, for the Oxford Nanopore technologies, the advantages here include the ability to sequence long reads, even longer than PacBio. And for the very first time, direct sequencing of the RNA and detection of RNA modifications, thus omitting the step of cDNA generation. However, sequencing with Oxford Nanopore is considered to be expensive, and the instrument has a high overall error rate, and large amounts of starting material is required. Now that we have seen briefly the history and the different sequencing tools that are currently available on the market, in the next couple of slides, I'm going to highlight the most commonly used applications of DNA sequencing. In this figure here, you can see a brief summary of those applications divided over two main categories, depending on whether we are interested in investigating or studying the genome or the epigenome. If our research aim is focusing on the genome, we can use DNA sequencing applications, including whole genome sequencing, that can be done with one of two approaches, either de novo or resequencing. In addition to whole genome sequencing, we can also perform whole exome sequencing, target region sequencing, and microbial sequencing if we are interested in microbial research. Microbial sequencing can be done with one of two approaches, either a more targeted amplicon-based approach or a more global shotgun-based approach. On the other hand, if our research aim is focusing on the epigenome, then we can rely on many DNA sequencing applications, including but not limited to bisulfite sequencing, ChIP-seq, cut-and-run, ATAC-seq, and many others. So starting with the first application, whole genome sequencing. By definition, whole genome sequencing is a technique that is used in order to sequence the entire genome of an organism at a single time. Whole genome sequencing is important because it provides us with a deep insight into the DNA sequence of different living organisms, such as humans, animals, plants, and microbes. One of the main advantages of whole genome sequencing is that it is able to identify and analyze different variants of the genome at a single base resolution. As I mentioned earlier, whole genome sequencing can be done in one of two approaches, either de novo sequencing or resequencing. With the first approach, called de novo sequencing, our aim here is to sequence a novel genome for an individual where no reference genome is available. By doing that, we are trying to assemble and annotate a reference genome for the very first time. So with the de novo sequencing approach, the DNA will be fragmented into smaller pieces, and then later, upon sequencing, the sequencing reads will be assembled to generate a reference genome that did not exist before. On the other hand, with the resequencing approach, we are trying to sequence a new example or a new individual of a species where the reference genome already exists. By doing resequencing, our aim here is to compare differences and variations between individuals or between individuals and the reference genome of the species that they belong to. So with the resequencing approach, the DNA will be fragmented into smaller pieces, and later upon sequencing and obtaining the sequencing reads, they will be mapped back to the reference genome that already exists in order to find variations and differences. Now, speaking of variations, there are different types of variants that can occur throughout the genome. Those can be as simple as single nucleotide variants affecting only one nucleotide at a time. Or they can be deletions or insertions affecting less than 50 base pairs at once. Deletions and insertions are commonly known as indels. Now, if the variants affect more than 50 base pairs at a time, they are commonly referred to as structural variants, and these include tandem duplication, inversion, translocation, copy number variants, and others. Now, DNA variants are actually very important because the process of variation itself contributes to differences between the individuals of a specific species. More importantly, depending on the location within the genome where the variation happens, these variants can have either no impact or severe consequences on the individual. Among the severe consequences that we might end up with include non-functioning or truncated proteins, impaired function of regulatory elements, or even disruption of cellular mechanisms. Now, moving on, another commonly used application of DNA sequencing is whole exome sequencing. By definition, whole exome sequencing is a technique that is used in order to sequence all of the protein coding regions of the genes within the genome. In fact, the protein coding regions within the genome are referred to as the exome. Whole exome sequencing as a technique is very important because even though the exome represents only 1% of the human genome, it harbors more than 85% of known disease-related variants. Our aim by performing whole exome sequencing is to identify genetic variants that alter protein sequences, including those that are responsible for Mendelian and polygenic diseases such as Alzheimer's. In comparison to whole genome sequencing, whole exome sequencing is considered to be very cost-effective and can be applied in both academic research and clinical diagnostics. For every whole exome sequencing experiment, there are two main steps. The first one is exome enrichment, where we try to enrich for the exome within the genome by using commercially available kits such as those provided by Agilent, Twist, or ITT. After enriching for the exome, we can proceed with DNA library preparation and sequencing using Illumina, PacBio, or any other instrument of interest. In addition to whole genome sequencing and whole exome sequencing, another commonly used DNA sequencing application is target region sequencing. With target region sequencing, we are now focusing on sequencing only a selected set of genes or genomic regions that we think is involved in a specific disease or a phenotype. The advantages of target region sequencing over whole genome sequencing and whole exome sequencing is that it requires less sample input and will eventually produce a smaller data set that might be easier to analyze and interpret. Target region sequencing can be done in different methods. One of those is called hybridization capture or target enrichment, and the other is called amplicon sequencing. Hybridization capture methods are recommended if you are interested in studying more than 50 genes or more than 50 genomic regions at a time. The advantages here are that The experiment itself will be more comprehensive. However, it requires longer hands-on time. Hybridization capture can be used using commercially available kits, such as those provided by Agilent, Novogene, or Roche. And it has been used for different research areas, including exome sequencing, genotyping, oncology, gene discovery, and many others. On the other hand, amplicon sequencing is recommended when we have a smaller gene content or genomic regions set. So we talk here about less than 50 genes or genomic regions. The advantages of amplicon sequencing over hybridization capture include cost, so it is more affordable and has easier workflows. Some of the research areas that really benefit from amplicon sequencing include genotyping by sequencing, detecting CRISPR editing events, or disease-associated variants. Now, moving back to the hybridization capture methods and focusing on oncology, we know that DNA sequencing for the last decades have been very important to provide us with a high resolution and a global view of the cancer genome. And this is why a lot of different targeted oncology panels were developed in order to deliver a comprehensive profiling test for various cancers. One of those oncology panels that is available on the market is NovoPM2. NovoPM2 can be used in order to provide us with a profiling test for solid tumors. The oncology panel itself analyzes 484 genes that have been shown to be clinically relevant for the diagnosis and the treatment of various solid tumors. In addition to the profiling of these 484 genes, the oncology panel itself can also provide us with the analysis and tailor the treatment recommendations for cancer patients. Now, moving on to different applications of DNA sequencing, we arrive to metagenomics. So metagenomics, by definition, is the study of microbial communities in their original habitats. And it is very important because it provides us with a comprehensive insight into the interactions within microbial communities. As I mentioned earlier, metagenomics can be done in one of two approaches. The first one is called targeted metagenomics, or commonly known as sequence amplicon-based approach. Using targeted metagenomics, we focus our sequencing on a selected set of genes or markers that we know are present within the microbial community. However, with whole metagenomics or shotgun whole metagenome sequencing, we have a global approach. And with this, we are sequencing all of the DNA material that is present within the microbial community. And finally, one of the main research areas that really benefits from DNA sequencing is epigenetics. By definition, epigenetics is the study of stable phenotypic changes that do not involve alterations in the DNA sequence itself. The epigenetic system has two main characteristics. The first one is heritability. In other words, the epigenetic system is mitotically and or meiotically heritable. So epigenetic information can be faithfully transmitted from the mother cell to the daughter cells or from the parents to their offspring. In addition to heritability, the system is also reversible. So any epigenetic information that is present at this point in time can be later reversed in the future. For those of you that are interested in using DNA sequencing for epigenetics, there are a lot of different applications that are available depending on the research aim. If you are interested in investigating DNA methylation, then we can rely on applications such as whole genome bisulfite sequencing, or WGBS. For those of you that are interested in investigating histone modifications and transcription factors, then we can rely on methods such as chromatin immunoprecipitation sequencing, or ChIP-seq. And finally, if we are interested in investigating chromatin accessibility or chromatin conformation, then we can use methods such as ATAC-seq or HiSeq. So to recap what I have showed you so far, there are a lot of DNA sequencing applications available that you can choose from depending on the research aim and whether you are interested in investigating the genome or the epigenome. Now, to move on, in the next couple of slides, I'm going to highlight some of the most commonly used applications of RNA sequencing. So RNA sequencing, by definition, is the process that determines the nucleic acid sequence within the RNA molecule. almost all of the RNA sequencing experiments will reveal two main pieces of information. The first one is the presence, and the second one is the quantity of the RNA in the biological sample at a given point in time. Among the most commonly used applications of RNA sequencing, we have gene expression level, isoform expression level, or even differential expression levels if we are trying to compare different experimental conditions to each other. Other applications where we can really benefit from RNA sequencing include gene fusions, RNA kinetics, variant discovery, alternative splicing, or even genome annotation. RNA sequencing is very versatile, and there are a lot of different research areas that really benefit from RNA sequencing applications, including but not limited to disease, diagnostics, therapy, biomarker, and even RNA delivery studies. Now, to better understand the different applications of RNA sequencing that are available and that you can benefit from, it is very important to understand the complexity of the different RNA species molecules that are present within the cells. So generally speaking, RNA can be divided into two main categories. The first one is the messenger RNA that is responsible for encoding for proteins. Almost all of the messenger RNA within the eukaryotic cells contains a poly-A tail. Everything else is considered to be a non-coding RNA. Non-coding RNAs themselves can be divided into two main types depending on their function. The first one is housekeeping non-coding RNAs, including ribosomal RNA that is involved in the process of protein translation, and tRNAs that are involved in the process of transcription. In addition to the housekeeping non-coding RNAs, we have regulatory non-coding RNAs that can be further subdivided into two main types depending on their structure. Those can be either circular RNAs or linear RNAs. Linear RNAs can be further subdivided into two main types depending on their length. On one hand, we have long non-coding RNAs that are longer than 200 nucleotides, and small non-coding RNAs that are shorter than 200 nucleotides. Small non-coding RNAs include microRNAs, siRNAs, piRNAs, and many others. In this table here, I tried to summarize the most commonly used assays for RNA sequencing. And as you can see, we really need to focus on the target RNA that we are interested in. So if we are interested in investigating the messenger RNA within the cells, then we can use an assay like messenger RNA sequencing. And if you remember, I mentioned earlier that the messenger RNA has a poly-A tail. And this is what we use in order to enrich for the messenger RNA within our sample. However, if our starting material comes from a fixed tissue or a prokaryotic sample, then we can use methods such as ribosomal RNA depletion. If we are interested in investigating messenger RNA and long non-coding RNA at the same time, then we can use assays like long non-coding RNA sequencing. And for this method, we rely on ribosomal RNA depletion. If our research aim is focusing on investigating small non-coding RNAs, then we can use small RNA sequencing. And for this assay, we rely on size selection, since almost all of the small non-coding RNAs are very short in length. For circular RNA sequencing, for circular RNA studies, we can use circular RNA sequencing. And for this method, we rely on ribosomal RNA depletion, and more importantly, linear RNA depletion, because we want to get rid of everything that is linear and enrich only for the circular RNA. And finally, if our research aim is to investigate all the different types of RNAs that can be present within the sample, then we can use approaches like total RNA sequencing or whole transcriptome sequencing. And for this, we rely on ribosomal RNA depletion and size selection. Now you might ask the question, what are the differences between poly-A enrichment and ribosomal RNA depletion? In particular, what are their advantages and disadvantages? So starting with poly-A enrichment, it is the most commonly used method for library preparation for RNA sequencing experiments. The principle of poly-A enrichment relies on oligo-DT primers that are able to recognize the poly-A tail of messenger RNA. And because of that, we can end up with an efficient enrichment of protein-coding polyadenylated RNA transcripts. One of the main advantages of using poly-A enrichment is cost, it is considered to be very cost effective. However, poly-A enrichment has its shortcomings, so it performs poorly for degraded RNA or fixed tissue samples, and more importantly, it cannot be used if our starting material comes from a prokaryotic sample. On the other hand, ribosomal RNA depletion relies on ribosomal RNA removal by using oligos that are complementary to ribosomal RNAs. And since we just remove the ribosomal RNA from the sample, we are now able to detect small and non-polyadenylated RNAs, in addition to the detection of long and shorter transcripts. So there is no 3' bias where the poly-A tail is usually located. The advantages of using ribosomal RNA depletion over poly-A enrichment include better performance on degraded RNA and fixed tissue samples, and more importantly, it is suitable and recommended for prokaryotic samples. However, one of the disadvantages of ribosomal RNA depletion is that more sequencing depth is required eventually. In addition to poly-A enrichment and ribosomal RNA depletion, we also need to take into consideration the differences between short-read and long-read sequencing platforms. So one of the issues that might come up is that short-read sequencing is not able to resolve complex isoforms that might arise during the transcription of a specific gene. This issue can be easily solved with long-read sequencing because now we are actually able to recover the full length of the individual transcripts, and thus we end up with more reliable isoform information. Now, talking about the differences between short reads and long reads sequencing, I think it is very important to see the advantages and the disadvantages of the different sequencing platforms if we are interested in an RNA sequencing experiment. So in this table here, you can see the differences between short read and long read sequencing in terms of RNA sequencing. Starting with Illumina RNA sequencing, the advantages here include very high throughput. The biases and the error profiles are very well understood, so we can always take that into consideration while we are performing data analysis. In addition to that, the computational workflows and pipelines are abundant and are available everywhere. More importantly, sequencing with Illumina for RNA sequencing is compatible with degraded RNA. However, sequencing RNA with Illumina has its disadvantages, including sample preparation, adding biases to all methods. And as I mentioned earlier, isoform detection and quantification can be very limited. And if we are interested in discovering new transcripts, then this would require a de novo transcriptome alignment or complex transcript analysis that can be very limited. In terms of applications, almost all of the RNA sequencing methods and applications have been developed to be compatible with short-read cDNA sequencing with Illumina. On the other hand, sequencing RNA with third-generation sequencing platforms, including PacBio and Oxford Nanopore, the advantages here include the ability to capture full-length transcripts, and the computational methods are simplified. However, sequencing with third-generation sequencing tools can suffer from low to medium throughput, sample preparation adding biases to many methods, and more importantly, these platforms are not recommended if our starting material has degraded RNA. In terms of applications, long-read sequencing using PacBio and Oxford Nanopore is highly recommended if you are interested in investigating isoforms or performing de novo transcriptome or any type of complex transcript analysis. So now that we have covered some of the most commonly used applications of RNA sequencing, in the next couple of slides, I'm going to guide you through the next generation sequencing workflow. This is an overview of the workflow that consists of 4 main steps. The first one includes sample preparation, where we are going to extract the DNA and the RNA from the samples that we are investigating. After that, we move into library preparation to prepare the DNA or the RNA library. And after that, we move into sequencing using the instrument of choice, followed by data analysis and data interpretation. So for any successful next generation sequencing project, whether it is DNA or RNA, it is very important to make sure that our starting material is of good quality. In order to check the quality of the extracted DNA or RNA, we can actually run an agarose gel. In the case of DNA, good quality DNA would look like a sharp, high molecular weight band on an agar-rosy gel, and we definitely need to avoid any scenarios where the DNA is degraded. A similar profile should be obtained with the fragment analyzer, where we expect a high molecular weight DNA and without any degradation. For the RNA, on the other hand, by running an agar-rosy gel, good quality RNA should give us two sharp bands representing the 28S and the 18S ribosomal RNA without any degradation. A similar profile should be obtained with the fragment analyzer, where we would expect to have sharp, defined peaks representing the 28S and the 18S ribosomal RNA without any degradation. In addition to the quality, it is very important to make sure that we have enough amounts of nucleic acids. And for this, we can use instruments like the nanodrop and the qubit to precisely quantify the amount of DNA and RNA that we have in our sample. Now that we have made sure that we have enough RNA and DNA of very good quality, we can proceed into the next step, which is library preparation. In this figure here, you can see the most commonly used library preparation workflow for samples that will be sequenced using the Illumina platform. So our starting material here will be double-stranded DNA in case of a DNA sequencing experiment, or it will be the cDNA that we have generated for an RNA sequencing experiment. After obtaining the double-stranded DNA or the cDNA, we proceed with fragmentation to generate short fragments that are compatible with the platform. After fragmentation, we perform end repair and A-tailing, followed by the ligation of the sequencing adapters that would make the DNA fragments compatible with the platform itself. After the ligation of the sequencing adapters, we perform multiple rounds of PCR amplifications in order to have enough material that we can use for sequencing. For sequencing with PacBio, after obtaining the DNA or the cDNA, we can ligate a specific type of adapters referred to as the SMRT smart bell adapters. And eventually, upon sequencing, these adapters will be recognized by the DNA polymerase that will proceed in sequencing the DNA or the cDNA that we have added. On the other hand, for sequencing with Oxford Nanopore, our starting material here can be DNA or RNA. The first step is size selection, plus an optional fragmentation if it is needed. After that, we can proceed into end prep and nick repair, followed by the ligation of the sequencing adapters before loading into the instrument and sequencing. Now that the library has been prepared, we move into the sequencing of these samples. One of the most important questions that we need to ask ourselves at this point is, how much should I sequence the samples? Now, when we talk about sequencing and data requirements, there are a lot of different factors and parameters that we need to take into consideration. And these include the research aim and the application that we are using, the size of the genome of the species that we are investigating, along with the genome complexity and how many repetitive regions we have within the genome. In addition to that, we also need to take into consideration the error rate of the sequencing instrument and the methodology that we are using. One of the most commonly used terms when we talk about sequencing and data requirements is the depth of coverage. So the depth of coverage represents how strongly is the reference genome being covered by the sequenced fragments. In this figure here, the depth of coverage is represented on the y-axis, and it is usually expressed as a value of 1x, 2x, 3x, or more. To make it easier during the calculations of data requirements, we can use the following formula to calculate the depth of coverage. So we can take the multiplication of the read length by the total with the total number of reads that we want to obtain at the end of sequencing, and we divide that by the genome size of the species that we are investigating. Here you can see a summary of the recommended data requirements for different applications that we have collected based on our experience at Novogene, but also on the reported literature. So for short read sequencing applications, if you are interested in de novo assembly for whole genome sequencing, then we recommend at least 100x coverage. For variant detection using whole genome sequencing or whole exome sequencing, we recommend something between 20 to 50x for frequent variants and more than 100x for rare variants. And finally, if the research aim is population studies using short read sequencing, then we recommend something between 20 to 50x. In parallel, if we are performing DNA sequencing using the long read sequencing platforms, then we recommend at least 50x coverage for de novo assembly using whole genome sequencing, and at least 10x coverage for large structural variant detection with whole genome sequencing. In case of frequent variants, then we recommend a coverage between 20, ranging from 20 up to 50. In addition to the DNA sequencing applications, it is also important to take into consideration the data requirements for RNA sequencing experiments. Here, there are different factors that we need to take into consideration, including the size of the genome of the species that we are investigating, and also the research aim. So if we are interested in differential expression, in comparing the gene expression profiles between different experimental conditions, then we recommend at least 5 million sequencing reads if the samples come from a species with a small genome, like bacteria or fungi. However, if the samples come from an organism that has a larger genome, like a human or mouse, then we recommend something between 15 to 20 million reads for differential gene expression. If our research aim is to detect rare transcripts and discover transcripts that were not reported before, then we need to generate at least 50 to 65 million reads if the samples come from a species with a small genome, like bacteria or fungi, or something between 100 up to 200 million reads if the samples come from organisms with a large genome size, like human or mouse. So now we come back to the workflow. And at this point, what we have done is basically sequencing the samples that we have prepared. The next step, naturally, would be data analysis. I'm not going to go into details about the different data analysis pipelines that you can use, as this has already been covered by previous webinars, and there will be more webinars this year talking in details about the different DNA and RNA data analysis pipelines. But just to give you an overview of the main steps of the data analysis pipelines. So the first step for data analysis is data QC or data quality control. It is very important to make sure that the sequencing reads that we obtain from the sequencing instrument are of good quality. So the first step is to perform data QC. The input will be the raw data that we obtained from the sequencing instrument, and the output would be clean data that can be used for downstream analysis. The second step in the pipeline will be alignment to the reference genome of the species that we are investigating. And in addition to that, it is very important to remove any duplicates that might have come during the PCR amplification steps of library preparation. These duplicates are considered to be an experimental artifact during library preparation, and they need to be removed before any downstream analysis. Once we have the clean data, after removing the duplicates, we can proceed with either variant calling, annotation, or other types of analysis if we have performed a DNA sequencing experiment, or we can now start investigating differential analysis, enrichment analysis, or others if we have performed an RNA sequencing experiment. There are a lot of different types of advanced analysis that can be used depending on the research aim. And once this analysis has been performed, then we can proceed into data visualization and data interpretation to make sense of the sequencing data that we have generated. So now that I have guided you through the main steps of the sequencing workflow, I arrived to the last part of my webinar. And in the next couple of slides, I'm just going to show you some examples of what we can expect for the future of next generation sequencing. So I mentioned earlier in my introduction, the human genome. I think it is very important to put the human genome in perspective here and see how much progress we have actually achieved in the last 20 years. So we know that the first draft of the human genome was done in the early 2000s, and that came with a price tag of $300 million, and it took at least 10 years to complete. Now, 20 years later, you can see the improvements that have been happened because now we are actually able to sequence the human genome for around $1,000, and it would take us only 24 hours. So there have been a lot of developments and advancements in the field of next generation sequencing during the last 20 years. It is important to mention here that, that when the first draft of the human genome was developed, we were actually able to obtain for the very first time a high quality human genome sequence. But one of the issues there is that it accounted only for 92% of the human genome. The remaining 80% were not readable because of the limitations of the technologies that were available at that time. However, these 8% of the genome that were not readable are very important for structural and medical reasons. And it took us around 20 years since the first draft of the human genome to fully and truly sequence the human genome, and that was published in 2022. In addition to the human genome and the advancements in technologies, there have been a lot of advancements for specific types of applications. And one of those that it is very important to mention include performing RNA sequencing at a single cell resolution. So during the early days of performing RNA sequencing, scientists and researchers were performing bulk analysis on a tissue that is made up of different types of cells. And then in the end, what we obtained was actually an average gene expression from all of the different cells that are present within the pool. And we were missing on information, including cellular heterogeneity. In order to overcome this issue, lots of different techniques were developed, and those tried to focus on generating gene expression profiles from individual cells within. And with this, now we were actually able to understand the heterogeneity and discover different subpopulations of cells that are present within the pool. And this has been due to the successful technologies that are provided by many companies, including 10x Genomics, NanoString, and Parse Biosciences. Another trend that we also need to take into consideration, and this trend has been gaining a lot of traction in the last couple of years, is multiomics. So by definition, multiomics refers to the combined study of multiple types of omics data, including genomic, transcriptomic, epigenetic, proteomic, and even metabolic datasets. By performing multiomics, our aim here is to better understand the relationships and the interactions between these different data sets. So now it is not enough to just focus on the genome and forget about the epigenome or the transcriptome or the proteome. Living organisms are very complex, so it is very important to understand the relationships and the interactions between all of these different data sets. And with this, we're actually able to comprehensively understand biological systems and use the information to investigate and study different diseases, development, and even evolution. Now, focusing on 2023 and what we can expect for the next for this year, I think there are some announcements that have been made recently, which are very important. I will start first with Illumina. So recently, Illumina launched the new NovaSeq X series of instruments. And with this, we are now able to bring down the price tag of sequencing the human genome from $1,000 down to approximately $200 per genome. And the platform has a much better performance, so it can generate 2.5 times the output compared to the previous generations. In addition to investing in short-read sequencing, Illumina has been heavily investing in long-read sequencing, and now we will be able to sequence beyond the 500 base pairs limitation up to 10 kb. In addition to improvements in the platforms themselves, Illumina has also been investing in improving the chemistry and also expanding into the clinical research. Along with Illumina, Pacific Biosciences, or PacBio, has also been heavily investing in improving their platforms and their technologies. They recently announced the new instrument called Revio for long-read sequencing that has a 15 times increase in throughput compared to the previous generations. Using the Revio platform, we are now able to sequence 1,300 human genomes per year, and that came also with a reduction in the sequencing of the human genome using long-read sequencing down to around 1,000 In addition to the new instrument, PacBio also invested in improving and simplifying sample preparation protocols and also in the field of single-cell long-read applications. In addition to long-read sequencing, PacBio is now entering the short-read sequencing market using the ONSO instrument that they recently launched, and there will be more details about it in 2023. And with this, I come to the end of my webinar, and I'm happy to answer any questions that you might have.
Thank you so much, Fred. That was a great presentation. I think everyone can agree, very much clear. Let's dig into a couple of questions. We do have just 5 minutes before the webinar ends. So I will just try to get some of the questions that has been asked through the Q&A tag. So the first questions, it would be from Mikhail, that is asking how to get rid of host DNA in whole genome sequencing data analysis.
Yeah. So the hosted genome can be the hosted genome data can be eliminated during different softwares and pipelines that are available for data analysis. I don't want to go too much into the details here, but this will definitely be covered in future webinars focusing mainly on microbial research. But to keep it brief, because we're running out of time, I would say that we are actually able to know the sequences of the hosted genome, and that can definitely be eliminated during data analysis and enriching only for the pathogen genome.
Great, thank you so much. So let's dig into the next questions. It's from Michael and is asking, why is duplicate removal important? Does it do anything more than slow down analysis and increase that amount?
Yeah. Duplications can arise during the PCR amplification steps of library preparation. And this is something that we cannot omit. So PCR amplification is an important part of library preparation. And later when we do the DNA, the data analysis, it is important to remove them because they are considered to be an artifact and they might create a bias. So suddenly we might see now some genes more expressed in an RNA sequencing experiment, but this is not real. This is just an artifact because some regions have been duplicated during the PCR steps. So to get a real idea about what is actually happening within the data that we have sequenced, it is very important to remove those duplicates.
Great, perfect. Let's get into the third question. This is from Anna, and she's asking, in your opinion, PacBio can substitute Illumina to bacteria species identification in microbiome study. Diversity is correctly detected or underestimated?
Yeah, there have been a lot of developments for microbial sequencing with long read sequencing, and now we are actually able to do, for example, 16S or 18S or ITS sequencing for microbial projects using PacBio, and not only targeted but also shotgun metagenome sequencing using PacBio. So the methods are there. The data analysis pipelines are available, so we can definitely look into the different elements of the DNA sequencing, including species discovery and annotation and much more. And this has been developing in the last couple of years, and I expect this year and next year to even have more developments in terms of pipelines and data analysis and how we can actually interpret the data.
Okay, great. I think we have just space for one last questions before we're closing up. So Fritz is actually asking, what is the cheapest way to analyze genetic diversity of an ancient DNA?
Yeah, for ancient DNA, the cheapest way will be to use a targeted approach. So if there is a selected set of markers or genes or genomic regions that we think is available, then we can just need to amplify those regions and use a targeted approach for sequencing. If it is unclear, the regions that you are interested in, then I would recommend using a more global approach. And that can be more expensive because now we are sequencing more regions and more data needs to be generated. And that will also complicate data analysis because we have now a larger data set that was generated. So to keep the cost down, if we have the information already about the genes or the genetic regions of interest, then we can definitely use a targeted approach or amplicon-based.
That's great, Red. Thank you so much for answering all these questions. I think thank you all for your time today. We hope you enjoyed the webinar and found it useful. Thanks for all your questions today. As mentioned, we haven't managed to answer all your questions live, but we will get back to you via email next week with all answers. Please, in the meantime, we will email you the link to the webinar recording and the slide deck, along with a short survey that we would appreciate you taking time to complete. The survey will help ensure we'll make future webinars even more relevant to you and your research goals. There will be a link in the following up emails for you to view a certificate of attendance if you need one. I saw that actually in the chat there was to a lot of people that were requesting it, so don't worry, we will send it out. In the meantime, please stay in touch. If you have any upcoming NGS project now, it will be a great time to contact us. You can follow us on Twitter, on LinkedIn, or you can get in touch via our website at novagene.com. Links are here in the description. We look forward to seeing you all on the next webinar. So thank you again. Thank you so much, and see you next time.